Ligation can be defined as the act of joining, and in biology, the term refers to an enzymatic reaction that joins two biomolecules with a covalent bond. This video describes the application of DNA ligation in molecular biology research. In the cell, DNA ligases are enzymes that identify and seal breaks in DNA by catalyzing the formation of phosphodiester bonds between the 3' hydroxyl and 5' phosphate groups of the DNA backbone. Ligation occurs during normal cellular processes such as DNA replication to repair single and double-strand DNA breaks. In the laboratory, DNA ligase is routinely used in molecular cloning, a process that joins endonuclease-digested DNA fragments, or inserts, with an endonuclease-digested vector, such as a plasmid, so that the fragment can be introduced into host cells and then replicated. Endonuclease digestions involve the use of restriction endonucleases, or restriction enzymes, which create nicks at specific stretches of DNA. These nicks can resemble single-strand breaks producing 3' and 5' overhangs called sticky ends, or double-strand breaks with no overhangs called blunt ends. Ligating sticky ends is advantageous because the complementary overhanging base pairs stabilize the reaction. Because blunt end ligations don't have any complementary base pairing, the ligation is less efficient and more difficult for the enzyme to join the ends. Sticky and blunt ends cannot, under normal circumstances, be ligated together. However, the Clenau fragment, the product of DNA polymerase I digested with subtilisin, can convert sticky ends to blunt ends. Clenau possesses 3' to 5' exonuclease activity that chews up 3' overhangs, and polymerase activity that blunts 5' overhangs by extending the 3' end of the complementary strand. When the goal is to insert a gene into a plasmid, resealing of vector DNA, called self-ligation, is a common undesirable outcome for a ligation reaction. Alkaline phosphatase treatment of vector DNA post-digestion removes 5' phosphates on both ends and prevents this undesirable outcome. As we mentioned previously, vector and insert DNAs are digested with endonucleases prior to beginning a ligation. Following gel purification of digested vector and insert, DNA concentrations are measured on a spectrophotometer to determine the concentration of the purified vector and insert. From this concentration, the number of molecules of insert or vector in 1 microliter can be determined based on the average molecular weight for a DNA base pair and the number of base pairs in each fragment. Based on the calculated molecular concentration of vector and insert, a 3:1 ratio of insert to vector is calculated to determine the volume of vector and insert used in the reaction. This 3:1 ratio of DNA insert to vector is desirable because it ups the probability of the insert being ligated into the vector versus the vector ligating itself. Now that we've determined the amount of vector and insert DNA to use in the reaction, we proceed to set up the ligation on ice. The order in which reaction components should be added to your microfuge tube is as follows. Sterile water, enough to make a 10 microliter final volume. In our case, we'll use 4 microliters. 1 microliter of 10x ligation buffer. 1 microliter of 10 millimolar ATP. 1 microliter of vector. 3 microliters of insert as calculated, and finally 1 microliter of DNA ligase. The reaction is mixed thoroughly, centrifuged, and incubated at the appropriate temperature. Whether you are doing a sticky or blunt end ligation impacts the temperature and duration of the ligation reaction. For example, a sticky end ligation with the 6 base pair overhang can be carried out near room temperature for about an hour because the complementary ends stabilize the joining of fragments. Short overhangs or blunt end ligations should be carried out between 14 to 20 degrees Celsius overnight. Now that we've learned how to set up a ligation reaction, let us now look at some of the applications of this procedure. Ligations can be used to directly insert PCR amplified fragments into linearized plasmids. Here you see a researcher taking a sample of frozen mouse brain, isolating genomic DNA from it, and then subjecting it to bisulfite PCR, which is a PCR-based method to detect methylated DNA. PCR products are then directly ligated into plasmid to create a library of genes that are methylated in that particular brain region. Ligations can be used to attach oligonucleotide linkers, which contain binding sites for PCR primers, to purify DNA fragments. When working with tumor samples, scientists can use this approach to sequence tumor genomic DNA with the hope of identifying tumor-causing mutations. In this video, ligation is performed on DNA isolated from formaldehyde-fixed cells and subsequently treated with the restriction enzyme in Clenau in the presence of biotin, which is then used to pull down ligated DNA. This DNA is then amplified using PCR and the product sequenced to identify chromatin interactions at various scales as shown. You have now learned about DNA ligase, various principles involved in setting up a ligation in the laboratory, potential problems and fixes, and various applications of ligation in molecular biology research. Thanks for watching.